Hello students, welcome to BIOZONE classes, and in today's session we discuss about the C4 cycle from class 11th chapter Photosynthesis in higher plants. From last few classes we already discussed about the C3 cycle and the different phases of photosynthesis. When we study about C4 cycle, so you mainly remember C4 cycle means we discuss about one of the phase of photosynthesis, that is the dark phase of photosynthesis. During dark phase, commonly we say the carbon dioxide fixation is take place, and this fixation of carbon dioxide is either take place by 3 carbon compound or by 4 carbon compound. As today we discuss about C4 cycle, so that means the CO2 fixation product, the stable product of CO2 fixation. So here you remember that C4 cycle means the path of carbon dioxide fixation where the stable product of CO2 fixation is 4 carbon compound. What is that compound that we study when we come towards the C4 cycle? But this compound is 4 carbon compound. Therefore, this path or cycle is called as C4 cycle. Secondly, you remember the C4 cycle is provided by the scientists Hatch and Slack. So here you remember the Hatch and Slack provide this cycle for CO2 fixation. So this part you remember. That's why this cycle is also called as Hatch and Slack cycle. This you remember. Now when we study about the Hatch and Slack cycle, so the plant where this cycle appear, this is one of the important question, that what are the examples of plant where the C4 cycle is present. So here you remember C4 plants. What are the C4 plant? So C4 plant means commonly maize, sugarcane and sorghum. For maize and sugarcane, repeatedly questions are asked in your competitive exams, so that you remember the maize and sugarcane as well as sorghum are the predominantly C4 plant, and these plants show the pathway for CO2 fixation, that is Hatch and Slack cycle. What is the identical property of these plants? So here you remember C4 plants show a special anatomy in their leaf, that is called as crange anatomy. Related to crange anatomy, separately I already uploaded one video that what is crange anatomy. So I will provide a link in description, so you can go through that link and clear your doubt regarding the crange anatomy. Crange anatomy, here you remember, in crange anatomy whenever we study the crange anatomy, that the cells are arranged layer by layer, as well as crange anatomy is a feature of leaf. First thing you remember, and the leaf of these plants show the dimorphic chloroplast or dimorphism in chloroplast. So now what is dimorphism in chloroplast? Dimorphism in chloroplast means a 2 type of chloroplast is present in the leaves of C4 plant, that the first chloroplast is present in mesophyll, so that is commonly called mesophyll chloroplast. How this chloroplast is differ from bundle sheath chloroplast? The second type of chloroplast is bundle sheath chloroplast. This part you remember. Now the mesophyll chloroplast you remember, it is granule chloroplast or normal chloroplast that we study. The grana and stoma both are present, but in bundle sheath chloroplast, only stroma is present, grana is completely absent. That's why it is called as agranule chloroplast. This part you remember very well. Now when we come towards the CO2 fixation pathway, so in mesophyll chloroplast or in mesophyll cells, you remember only the C4 path or C4 cycle is take place. So CO2 fixation in mesophyll cells take place by C4 cycle. For objective purpose, if question is asked that the C4 cycle or CO2 fixation is take place in, so C4 cycle begins in mesophyll cells as well as the CO2 fixation, that is the production of 4 carbon compound, is also take place in the mesophyll cells of the C4 plant. Now when we come towards the bundle sheath cells or bundle sheath chloroplast, so in bundle sheath chloroplast you remember C4 as well as C3 cycle of CO2 fixation is appear. This part you remember very well because the enzyme Rubisco already we studied in C3 cycle. The enzyme Rubisco is required for C3 cycle. That Rubisco is present in the stroma of bundle sheath cell. Rubisco is completely absent in mesophyll chloroplast, and because of that the C4 cycles only appear in mesophyll, whereas in bundle sheath along with C4, C3 cycle is available. And this is the reason that whenever question is asked that which plants are having high efficiency for CO2 fixation, whether C3 or C4, so you remember very well the plants, that is C4 plant, have high efficiency for CO2 fixation because along with C4 they show the C3 cycle. So this part you remember. And now I come towards the cyclic path of the C4 cycle. So here we come towards the cyclic path. Here I draw the 2 cells. This is the upper cell, that is the mesophyll cell. This mesophyll cell you remember, the mesophyll cell is connected by bundle sheath cell with the help of plasmodesmata or cytoplasmic strand. So here we draw the plasma membrane. Because the chlorophyll is present in mesophyll cell, that's why you go through the NCERT. So in NCERT, the color of mesophyll cell become green, whereas the bundle sheath cells are colorless because the chlorophyll is completely absent in bundle sheath cell. So this upper cell here we draw, that is the mesophyll cell, and this is the bundle sheath cell. This you remember. Now here I just draw the plasmodesmata or cell to cell connections, and now here you remember the first carbon dioxide acceptor. The first carbon dioxide acceptor for C4 cycle is phosphoenol pyruvate, or commonly it is called PEP. So this phosphoenol pyruvate you remember is the first CO2 acceptor. This is a 3 carbon compound. This 3 carbon compound absorb carbon dioxide, and the important class of enzyme is PP carboxylase or already we studied the PPKs. Commonly this is called as PPKs. So PP carboxylase or PPKs is an important class of enzyme. This enzyme responsible for the fixation of carbon dioxide and the 4th carbon stable product that I earlier mentioned in C4 cycle. The first stable product of CO2 fixation is 4 carbon compound. That 4 carbon compound you remember is oxaloacetic acid. This oxaloacetic acid is the first stable product. Oxaloacetic acid is now convert into malic acid in NCERT. Instead of providing the name, only the C3 acid and C4 acid is provided. C3 acid means phosphoenol pyruvate, C4 acid means oxaloacetic acid. This oxaloacetic acid get convert into transportable form, that malic acid. This malic acid also 4 carbon compound, and then after the malic acid from mesophyll cell transported towards the bundle sheath cell. In bundle sheath cell, you remember, in bundle sheath cell, this malic acid now undergoes in decarboxylation process, and because of that again here the decarboxylase enzyme is activated, and in presence of decarboxylase enzyme, carbon dioxide is releases from the malic acid, and again the 4 carbon compound become convert into 3 carbon organic acid, that is pyruvic acid. So pyruvic acid is a 3 carbon organic acid, and the carbon dioxide which is releases due to decarboxylation, this carbon dioxide is used by RUBP. RUBP is a CO2 acceptor for C3 cycle, and again this RUBP involve in C3 cycle and result in the production of glucose. So in bundle sheath along with C4, C3 cycle is take place. In mesophyll only and only C4 cycle is take place. Now this pyruvic acid or C3 acid again transported to mesophyll cell, and here this pyruvic acid undergoes in regeneration of phosphoenol pyruvate. In regeneration of phosphoenol pyruvate, you remember ATP is required. This ATP broken down into ADP and inorganic phosphate, and the inorganic phosphate used by pyruvic acid in the formation of phosphoenol pyruvate. So this part you remember very well, that how the C4 cycle is take place in mesophyll cell as well as in bundle sheath cell. So this was all about for C4 cycle. Now when you go for preparation of competitive exam, just that is NEET only. So one thing you remember, some important questions are repeatedly asked, that what is a stable product. So oxaloacetic acid you remember is a stable product. What is a carbon dioxide acceptor? CO2 acceptor is a 3 carbon compound phosphoenol pyruvate. The enzyme which responsible for carboxylation, so PP carboxylase or peptase. Secondly, the peptase is present in which cell you appear the peptase or PP carboxylase. So you remember mesophyll cells. The mesophyll cells are responsible for peptase or PP carboxylase. Okay, rubisco. The rubisco is present in bundle sheath cell, and because of the presence of this rubisco in bundle sheath cell, along with C3, the C4 cycle is regularly take place. So this much you remember for the C4 cycle. After that we come towards the comparison in next sessions. We first study about, maybe next session quite long, because we completed anyhow we try to sum up this chapter, because the factors of photosynthesis one topic is there, photorespiration is there, and after photorespiration comparison is provided in NCERT between the C3 and C4. So in next session, first we discuss about photorespiration, After that, the factors affecting factors affecting the phenomena of photosynthesis, because in that one important law is there, that is Blackman's law of limiting factor. So that we study first, and then after we discuss about the comparison between C3 and C4 cycle. And with this, in next session we will finish our chapter photosynthesis in higher plant. In case if you feel any doubts and difficulty in this chapter as well as in another topic of the biology from 11th and 12th, so kindly ask those doubts, clear your doubts, and up to next session, stay motivated and stay blessed.